sometimes a scientific paper doesn't just present results, it tells a story and how it was built, and that's exactly what happened with the study I want to walk through today. The conclusion wasn't even the most interesting part. What stood out was that the data had been sitting untouched in storage for nearly 40 years, carefully collected, fully recorded, and then never properly analyzed in a complete way. When someone finally went back and ran the numbers correctly, the outcome pointed in the opposite direction of what the field had believed for decades. That is where this begins, not with certainty, but with a question that was left unresolved for generations. The focus of this story is a single type of fat that exists almost everywhere in modern life. It is in home kitchens, it is used in restaurant fryers, and it shows up in the majority of packaged foods on supermarket shelves. It is so common that most people consume it daily without thinking about it. The concern is not about fear or blame, but about what happens inside the body when this substance is processed, heated, and consumed in large amounts over time. The mechanism being discussed is not a simple surface level effect. It is a chain of biological events that begins at the smallest functional level of the cell. And to understand it properly, you cannot reduce it to slogans or labels. You have to follow the steps as they actually occur inside living tissue. Before going deeper, it is important to clarify what this discussion is not. This is not an attempt to label one ingredient as the cause of every modern illness. That kind of narrative is usually too simple to be accurate and often leads people in the wrong direction. Instead, the goal here is narrower and more precise. There is a documented biochemical pathway in which a specific category of fat, when it is heavily processed, oxidized, and concentrated in the way modern food production makes it, can damage a critical structure inside cells. That structure plays a central role in one of the body's most important safety systems. This is where the discussion becomes more than nutrition and moves into cellular biology. Inside your cells, there are systems responsible for energy, repair, and quality control. Among the most important are the mitochondria. These structures are often described as energy producers, but that description is incomplete. They are also involved in deciding whether a damaged cell should repair itself, remain inactive, or be removed entirely. That removal process is called apoptosis, or programmed cell death. It is not a destructive process in the harmful sense. It is a controlled system that protects the body by eliminating cells that are no longer functioning correctly. If a cell accumulates serious damage, including DNA errors that could potentially lead to cancer, mitochondria help trigger its removal before it becomes a larger problem. This is the system we are really talking about. And so the central question becomes this: what if a common dietary fat consumed in large amounts over time does not simply affect cholesterol or energy levels, but instead interferes with the mitochondria in a way that weakens this protective mechanism? What follows from that question is not a simple answer, but a deeper investigation into how modern dietary advice was formed and how later research began to challenge parts of it. For about 60 years, nutritional guidance in many countries, especially the United States, was built around a clear idea. Saturated fat was considered harmful because it was linked to higher cholesterol levels, and higher cholesterol was associated with heart disease. As a result, people were advised to replace animal fats like butter, lard, and beef fat with vegetable-based oils. These oils, including soybean, corn, sunflower, safflower, and canola, were promoted as healthier alternatives. They did, in fact, lower cholesterol levels in many controlled studies. That part of the evidence was real and reproducible. But lowering a single laboratory marker is not the same as improving long-term survival or overall health outcomes. That distinction becomes critical when looking at what happened next, because decades later, researchers revisited old clinical trials that had never been fully analyzed or published in detail. What they found began to shift the way some scientists viewed these oils entirely. And that reassessment starts with a forgotten data set that would later change the interpretation of an entire nutritional theory. That reassessment begins with something that at first glance sounds almost like a historical accident in science. In 2013, a researcher named Christopher Ramsden and his colleagues published an analysis in the British Medical Journal that didn't come from a new experiment. It came from something far more unusual in modern research. They went back in time. They reopened the data from an old clinical trial known as the Sydney Diet Heart Study, which had been conducted in Australia between the late 1960s and early 1970s. The study included 458 middle-aged men, all of whom had already experienced heart problems. This was not a general population sample. It was a high-risk group where outcomes could be tracked more clearly. The design of the original trial was straightforward. One group of participants was instructed to reduce saturated fat and replace it with safflower oil, a vegetable oil extremely high in omega-6 linoleic acid. The control group continued their usual diet without major changes. At the time, researchers were focused on cholesterol levels as the main indicator of cardiovascular risk. And indeed, cholesterol levels did drop in the group consuming safflower oil. That result aligned perfectly with expectations and reinforced the dominant nutritional model of the era. But what had never been fully published or properly analyzed was the most important outcome of all: mortality. Ramsden's team recovered the original records and finally ran a complete analysis of who died and when. What they found was not a small or ambiguous difference. The group consuming the vegetable oil did not experience improved survival. In fact, they showed higher rates of death overall, including more cardiovascular deaths compared to the control group. This meant that an intervention designed to reduce risk factors had, in real-world outcomes, potentially increased risk instead. That result alone would have been enough to raise questions, but the strength of the finding was not just in the numbers. It was in how the data had been collected in the first place. The Sydney trial had been randomized, meaning participants were assigned to groups by chance rather than personal choices. This is important because it eliminates a major source of bias. It prevents the possibility that healthier individuals simply chose the better diet. Both groups were comparable at the start. Even more importantly, the data had been recorded in real time long before any modern controversy or reinterpretation existed. The researchers conducting the original study fully believed that vegetable oils would improve health outcomes. There was no incentive to suppress unfavorable results from a theoretical standpoint, at least not intentionally. The issue was that the full analysis was never completed or published in its time. When Ramsden and his team finally reconstructed the dataset decades later, they were working with raw historical evidence that had not been influenced by modern debates. That is part of what makes the finding scientifically significant. It is not based on reinterpretation of published claims, but on finishing an analysis that was left incomplete. The implication was difficult to ignore. Lowering cholesterol through dietary substitution did not automatically translate into longer life or reduced mortality. In this case, the expected benefit did not appear, and the outcome trend pointed in the opposite direction. And this was not the only data set that raised similar questions. In 2016, the same research group published recovered results from another large and influential study, the Minnesota Coronary Experiment. This trial was much larger, involving more than 9,000 participants, many of whom were housed in controlled environments such as state mental hospitals and nursing homes. This setting allowed researchers to carefully regulate dietary intake, making it one of the more tightly controlled nutrition studies of its time. The intervention was similar. Saturated fat was replaced with corn oil, another oil rich in omega-6 linoleic acid. Again, the expectation was clear. Lower cholesterol should lead to lower cardiovascular risk and reduced mortality. Once again, cholesterol levels did fall in the intervention group, but when the mortality data was fully recovered and analyzed, the pattern repeated itself. There was no clear reduction in death rates. In some subgroups, particularly older participants whose cholesterol dropped most significantly, the risk of death was actually higher. So now the picture included two separate large trials, both originally designed to support the same nutritional hypothesis, both showing cholesterol reduction and both failing to demonstrate improved survival when the full data was examined. The researchers themselves described the situation as a disconnect between the published record and the underlying evidence. Not fraud, not conspiracy, but a gap between what was measured, what was analyzed, and what ultimately entered the scientific literature. At this point, a deeper question emerges naturally. If lowering cholesterol through these oils does not consistently improve survival, then either cholesterol is not the full story or something else is happening alongside it that counteracts the expected benefit. That question becomes the turning point in the entire discussion because it shifts the focus away from blood markers and toward what is happening inside the cells themselves. And that is where the story moves from clinical trial, mitochondrial biology. That shift in focus changes everything because instead of stopping at cholesterol or population level outcomes, the investigation moves down to the cellular systems that determine whether a cell survives, functions normally, or is removed from the body when it becomes damaged. The attention turns toward the mitochondria, and to understand why they matter here, it helps to appreciate their scale and importance in a way that is easy to overlook when they are described as simple powerhouses of the cell. In reality, mitochondria exist in enormous numbers throughout the body. A single muscle cell can contain thousands of them, packed so densely that they can make up a significant portion of the cell's volume. Across the entire human body, there are on the order of trillions upon trillions of mitochondria working continuously every second of life. They are not static structures either. In healthy tissues, mitochondria are constantly being broken down and rebuilt. In some cells, especially in the heart, the entire mitochondrial population can turn over in a matter of weeks. This means your energy system is in a constant state of renewal where old components are replaced and new ones are assembled based on the materials available. That constant rebuilding is essential for survival, but it also introduces a vulnerability. Because if the materials used to build these structures are consistently altered, damaged, or chemically unstable, the new mitochondria will reflect that over time. Mitochondria are responsible for producing ATP, the energy currency that powers nearly every function in the body. But their role goes beyond energy production. They are also deeply involved in regulating whether a cell continues functioning or initiates cell self-destruction. This is where apoptosis becomes central again. Programmed cell death is not random damage or failure. It is a tightly regulated process that protects the body. A cell accumulates too much internal damage, especially DNA damage that could lead to cancerous behavior. Mitochondria help activate a controlled shutdown. The cell is then broken down and cleared away safely. This is a critical defense mechanism. Without it, damaged cells would persist, divide, and potentially develop into tumors or dysfunctional tissue. So mitochondria are not just energy suppliers. They are also decision makers in cellular quality control. This dual role becomes especially important when considering what happens if mitochondrial function begins to degrade. If their structure or internal signaling is disrupted, two major systems are affected at once: energy production and the ability to trigger proper cell death. This is where the earlier dietary discussion connects directly to cellular biology. If a common dietary fat, when consumed in large amounts and repeatedly heated or oxidized, produces byproducts that interfere with mitochondrial structure, then the consequences would not be limited to one system. They would affect both energy metabolism and cellular safety mechanisms. To understand how this could happen, it is necessary to look at the structure of the mitochondrial inner membrane, where much of this control system is physically located. Inside each mitochondrion, there is an inner membrane that is highly folded. These folds are not random. They increase surface area and provide space for the protein complexes that carry out energy production. These protein complexes form what is known as the electron transport chain, a step-by-step system that transfers electrons and ultimately produces ATP. This process is highly sensitive. Electrons must move in a controlled sequence, and any disruption can cause inefficiency or unintended chemical reactions. When the system runs smoothly, energy is produced efficiently, and cellular signaling remains stable. But when that membrane environment is disrupted, electron flow can become less controlled. Instead of clean energy transfer, small amounts of reactive molecules can be produced. Over time, this creates oxidative stress, which can damage surrounding lipids, proteins, and even mitochondrial DNA. The system can become self-reinforcing. Damage leads to inefficiency. Inefficiency leads to more reactive byproducts, and those byproducts create further damage. Within this inner membrane, one particular molecule plays a critical structural role. It is called cardiolipin, a specialized fat found almost exclusively in mitochondria. Cardiolipin helps anchor the protein complexes of the electron transport chain, keeping them properly organized and functional. Just as importantly, cardiolipin is involved in regulating cytochrome C, a protein that sits at the intersection of energy production and apoptosis. While it remains anchored, it participates in energy generation. But when released from the membrane, it becomes part of the signal that initiates programmed cell death. This means cardiolipin is not just structural. It is part of the switch that determines whether a cell continues living or is instructed to shut down. And because cardiolipin itself is made up of highly unsaturated fatty acid chains, it is chemically vulnerable to oxidative damage. This makes it a potential target in environments where oxidative stress is elevated. At this point, the discussion is no longer just about diet in a general sense. It becomes about how the molecular composition of what we eat might influence the stability of the very membranes that control energy production and cellular life or death decisions. And that is where the mechanism begins to tighten, because the next step is to examine what happens when these fragile membrane components are exposed to oxidative breakdown products over long periods of time. Once the focus shifts to membrane stability, the discussion becomes much more specific, because now the question is not just about energy production in general, but about how a single type of molecular damage can affect both energy flow and cellular safety systems at the same time. Inside the mitochondrial inner membrane, cardiolipin plays a central organizing role. It helps hold together the protein complexes of the electron transport chain, ensuring that electrons move in a controlled and efficient sequence. When this structure is intact, energy production is stable, and the system operates within safe limits. But this system is not built from inert material. Cardiolipin is made up of highly unsaturated fatty acid chains, which means its chemical structure contains multiple vulnerable points where oxidation can occur. This is not unusual in biology. It is part of normal membrane design. However, it also means that under conditions of increased oxidative stress, cardiolipin becomes one of the first targets of damage. To understand why this matters, it is important to recognize what happens when oxidative damage occurs in these membranes. Reactive oxygen species and related breakdown products do not simply disappear after formation. They interact with nearby molecules, especially those that are chemically vulnerable, like polyunsaturated fats. When cardiolipin is damaged, its ability to properly anchor the electron transport chain begins to weaken. The protein complexes that rely on it for structural support become less stable. This leads to small inefficiencies in electron transfer. On its own, a small inefficiency might not seem significant, but within a tightly regulated system, even minor disruptions can have cascading effects. When electrons do not move cleanly through the chain, some of them can escape the intended pathway. These escaped electrons can react with oxygen, producing additional reactive molecules. This increases oxidative stress within the mitochondrion itself, which can then damage more cardiolipin, further destabilizing the system. This creates a feedback loop where damage and inefficiency reinforce each other over time. The system does not fail suddenly. Instead, it gradually loses stability as small disruptions accumulate. Within this environment, another critical protein becomes relevant, cytochrome C. This molecule sits in a unique position because it is involved in both energy production and the initiation of apoptosis. When it is securely bound within the mitochondrial membrane, it participates in the electron transport chain. But when it is released into the cell, it acts as a signal that triggers programmed cell death. Cardiolipin plays a key role in keeping cytochrome C anchored in place. When cardiolipin is intact, cytochrome C remains part of the energy system. When cardiolipin becomes oxidized or damaged, its binding strength weakens and cytochrome C can detach more easily. This is where the consequences become biologically significant. If cytochrome C is released at the right time, it contributes to the removal of a damaged or dangerous cell. But if the system is destabilized, that release can become irregular. In some cases, cells may trigger death prematurely. In others, the signaling may become unreliable, failing to activate when it should. Both outcomes represent a breakdown in cellular quality control. On one side, excessive or misplaced activation of apoptosis can contribute to tissue loss, energy decline, and dysfunction in organs that rely heavily on mitochondrial activity, such as the heart and brain. On the other side, insufficient activation means that damaged cells that should be removed are allowed to persist. This second outcome is particularly important in the context of cancer biology. Cells in the body constantly accumulate minor DNA damage from normal metabolic activity. Under healthy conditions, these cells are identified and eliminated through tightly regulated mechanisms involving mitochondrial signaling. If that system becomes less reliable, damaged cells may survive longer than they should, increasing the likelihood that they continue dividing with accumulated errors. It is important to be precise here. This is not a single-step cause of disease. It is a change in the reliability of a protective system. The difference is subtle but critical. Instead of directly creating disease, it alters the probability that damaged cells are successfully removed. This is also where broader physiological patterns begin to appear. The same mitochondrial instability that affects apoptosis signaling can also influence energy production. When electron transport becomes less efficient, ATP output declines. Over time, this can contribute to lower cellular energy availability, which may manifest at the organism level as fatigue, reduced metabolic resilience, and slower recovery from stress. At the tissue level, these effects are most visible in organs with high energy demand. The heart, the liver, and the brain are especially dependent on stable mitochondrial function. This is why mitochondrial dysfunction is often discussed in relation to cardiovascular decline, metabolic disorders, and neurodegenerative conditions. The key point is that all of these effects trace back to the same structural vulnerability, the stability of the mitochondrial inner membrane and its lipid composition. When that membrane is repeatedly exposed to oxidative and chemically reactive breakdown products, its integrity can gradually decline. As cardiolipin becomes more oxidized, the electron transport chain becomes less stable, energy production becomes less efficient, and apoptosis signaling becomes less reliable. This is not an abrupt failure, but a slow drift in system performance over time. And once this idea is established, the next step is to identify where those oxidative stressors come from in a modern context and why certain dietary fats may increase the formation of these reactive breakdown products both inside and outside the body. Once the mechanism is understood at the level of mitochondria and membrane stability, the next question becomes unavoidable. Where exactly do these oxidative stressors come from in everyday life? And why does modern nutrition seem to increase exposure compared to earlier patterns of eating? The answer begins with the chemistry of the fats themselves, particularly a category of polyunsaturated fatty acids that are abundant in modern industrial oils. One of the most common is linoleic acid, an omega-6 fatty acid that is found in high concentrations in soybean oil, corn oil, sunflower oil, safflower oil, and similar seed-based oils widely used in food production. Linoleic acid is not inherently harmful. It is an essential fatty acid, meaning the body requires it in small amounts for normal biological function. In natural diets, it appears in relatively modest quantities, often within whole foods like nuts, seeds, and unprocessed plant materials, where it is bound within a complex structure and protected from rapid oxidation. The key difference in the modern context is concentration and processing. Over the past several decades, the amount of linoleic acid in the average diet has increased dramatically due to widespread use of refined vegetable oils in cooking, packaged foods, and restaurant preparation. Instead of being consumed in small, naturally embedded amounts, it is now consumed in large, isolated, and repeatedly heated doses. This shift matters because polyunsaturated fats contain multiple double bonds in their chemical structure. These bonds are reactive sites, meaning they are more susceptible to oxidation when exposed to heat, light, and oxygen. When oxidation occurs, the fat does not simply remain intact. It breaks down into smaller reactive compounds, some of which are biologically active and capable of interacting with proteins, lipids, and DNA. One of the most studied breakdown products of oxidized linoleic acid is a family of reactive aldehydes, including a compound known as 4-hydroxynonenal, often abbreviated as 4-HNE. This molecule has been extensively studied in free radical biology because of its ability to form stable adducts with cellular structures. Unlike short-lived reactive oxygen species, which often act quickly and locally, 4-HNE is more chemically stable. That stability allows it to travel within cellular environments and interact with targets that are distant from its original site of formation. It does not simply vanish after being produced. It persists long enough to modify proteins and interfere with normal cellular function. This is where its relevance to mitochondria becomes important. 4-HNE has a strong tendency to bind to proteins and lipids within the mitochondrial membrane, including components involved in the electron transport chain and structural lipids like cardiolipin. When this occurs, it can alter the shape and function of these molecules, reducing efficiency and increasing the likelihood of electron leakage. As electron leakage increases, more reactive oxygen species are produced internally, which further accelerates lipid oxidation. This creates a cycle in which oxidative stress leads to membrane damage, and membrane damage produces more oxidative stress. In parallel, there is another layer of concern, the condition of these oils before they are even consumed. Oxidation does not only occur inside the body, it begins during processing, storage, and cooking. Industrial seed oils are typically refined at high temperatures, exposed to oxygen, and stored for extended periods before use. They may sit in warehouses, on store shelves, and in kitchens for weeks or months. Each stage increases the likelihood of partial oxidation. The process continues when these oils are used for frying, especially in commercial settings where oil is heated repeatedly over long periods. Each heating cycle accelerates the formation of oxidative breakdown products, meaning that the oil used in some fried foods may already contain a significant load of reactive compounds before it ever reaches the consumer. This is an important distinction. The concern is not only what happens after ingestion, but also what chemical state the fat is already in at the moment it is consumed. Once inside the body, these partially oxidized lipids and their breakdown products can integrate into cell membranes, including mitochondrial membranes. Because membranes are constantly being rebuilt and remodeled, the composition of dietary fat directly influences their structure over time. If a large proportion of dietary fat is highly unsaturated and frequently oxidized, the resulting membranes may become more chemically vulnerable. Over time, this can increase susceptibility to oxidative damage within the mitochondria, particularly in environments where metabolic demand is high. This is where the connection returns to cardiolipin. Since cardiolipin is itself rich in unsaturated fatty acids, it is particularly sensitive to oxidative stress. When exposed to reactive aldehydes like 4-HNE and other lipid peroxidation products, its structure can be altered, weakening its ability to stabilize the electron transport chain and regulate cytochrome C. At this point, the discussion is no longer just about individual molecules. It becomes about system behavior over time. Small shifts in membrane composition, repeated exposure to oxidative byproducts, and continuous turnover of cellular structures can collectively influence how reliably mitochondria perform their dual role, energy production and cellular quality control. And once that reliability begins to shift even slightly, the long-term implications extend into multiple systems in the body, from metabolic efficiency to tissue aging and cellular resilience. This sets up the final question in the sequence. If these processes accumulate gradually over years, what does that mean in practical terms for health, diet, and the kinds of everyday choices that influence mitochondrial stability over time,